how sex develops. Now, in certain species, it's not all about the genetics. Sometimes it's about things like temperature and whatnot that will determine whether you're male or female. And so it's not just about what genes turn on what genes, but there can be environmental factors that can influence that in other species. But for us, it's primarily determined by the genetics. So the primary sex determination, this is the first stage of whether or not you become male or female. And the secondary sex determination, there's actually two main points in this. One happens during development, and then obviously the other one happens at puberty. So there's kind of two main stages of this, but it even goes beyond this when we start talking about male and female. We're going to talk about hermaphroditism today, as well as sexual identity and things of that sort, having to do with neurons and whatnot and what we know at this point. So let's look at primary sex determination again. We had only talked about the testes yesterday, so I'm going to review on that, and then we'll go through the ovaries. But that's really all primary sex determination is you have an undifferentiated gonad. We called it the bipotential gonad because it has the ability to go either way, either to the ovaries or to the testes. And ultimately, it comes down to whether you're XX or XY that will determine whether or not they develop into ovaries or they develop into testes. I am going to ask you heavily on the genetics behind this part because we understand this extremely well. Okay? So now in other species, there are situations where the development, again, is not having to do with males are not always XY. In fact, in chickens, they have two of the same sex chromosomes for the males and different sex chromosomes for the females. So it's not always that the Y chromosome in every species is the male determining chromosome. Okay? So just so you understand that it's not always, hey, every species has an XY for the males and an XX for the females or whatnot. In fact, we don't even use an X sometimes. We say ZZ or something else in terms of their sex chromosomes. So just so you're aware of that. I will talk about some of the genes that are universal between species as well. So remember that the gonads develop from the intermediate mesoderm along with the kidneys. So they really are developing side by side here. In fact, as we showed before, the kidneys do play a role in helping the male testes develop the tubules that are going to carry the sperm in its development. Now, at about 7 weeks or so, 6 to 7 weeks, you do not have any male or female development in terms of whether the gonad is going to become an ovary or a testes. But by about 8 weeks, that's when these genes start kicking in and taking the bipotential gonad and turning it into either the ovaries or the testes. There are other derivatives here that we'll talk about that are part of the secondary sex characteristics that will come about as a result of the differentiation of the gonads into either the ovaries or the testes. So let's talk about the testes again, just as a recap. The SARI gene, or the sex-determining region of the Y chromosome, which is what that gene refers to, is primarily just to turn on SOX9. SOX9 is an autosomal gene, so it's expressed in both males and females. In fact, SOX9 is expressed even at this stage of gonadal development. So SOX9 is one of those that is universally expressed in both male and female development up to a certain stage. However, in the males, SOX9 will persist because the SARI gene will trigger the autoregulation of this, and then this will continue to propagate itself. In fact, this is one of those genes that is found in pretty much every vertebrate species that is the male-determining gene. Even if the species has two of the same sex chromosomes, they find that the SOX9 gene is activated in males. So this, in terms of an evolutionary perspective, very well could be the universal gene, and then in humans we've got this one that initiates this, but in other species it's still the SOX9 gene that initiates the formation of the testes. So this is one of those genes that is found in a wide variety of species as a male-determining gene. So SOX9 does quite a number of things in the testes during the development. Ultimately, what's going to happen is the bipotential gonad is going to start developing the testes cords. And these testes cords will ultimately surround the spermatogonium. They'll eventually hollow out when puberty hits, in which the sperm will be able to mature and then be able to come in through the efferent ducts into the Wolffian duct, which eventually becomes the vas deferens, and then connects with the urinary system to be able to come out the urethra. So the spermatogonium undergo this differentiation process primarily because of the SARI gene initiating SOX9 and SOX9 initiating other genes that are necessary for the formation of the testes. Otherwise, these stem cells would go down a different path if it weren't for these genes changing their overall fate. So here's just kind of a summary of the testes pathway. The SARI gene is only on for just a little bit in order to activate the SOX9 gene. It's the SOX9 gene that does tons. One thing in particular right here, this is a key factor between the difference between testes and ovaries, is the blocking of beta-catenin. It's been shown that beta-catenin, if it is not blocked, will force the gonad to become an ovary. So one of the things about the SOX9 gene is it prevents ovaries from developing by blocking beta-catenin. In addition to that, it also activates other genes that are necessary to then say, okay, we're not making an ovary, now this is what you're going to make. Because in reality, it's been shown in a number of species that in the absence of these, the default mechanism is essentially female development. It still needs the proper genes to be able to take the bipotential gonad and turn it into an ovary, but without the intervention of these genes to stop it from becoming an ovary, it's already kind of pre-established to go down the fate of becoming female. Another important factor in primary sex development is it binds the gene anti-Müllerian factor, which will cause the Müllerian duct to degenerate, which is one of the main components of a lot of the secondary sex characteristics of female, such as the uterus and the cervix and so on and so forth. Promotes FGF9. This is another key factor. They showed that when they took ovaries and actually caused them to produce FGF9, they were able to get migration of cells from the mesonephros, which is part of kidney development, to migrate into the ovaries. And that doesn't normally happen. Ovaries don't have cells from the mesonephros migrating in to help in the overall development. This is only for testes development. So they showed that FGF9 is one of those that's absolutely necessary and sufficient to be able to cause these cells to migrate from the mesonephros into the developing gonad. It also helps in the proliferation of the Sertoli cells, which will become the testes cords that will surround the spermatogonium. So these are kind of the major players in how the bipotential gonad will become the testes. They've done a number of transgenic experiments where they took the SARI gene and they put it on the X chromosome and it developed into a male. And they took the SOX9 gene and they put it on the X chromosome and further activated it, and it developed into a male. So they did a number of transgenic experiments where they would take some of these key genes and they were able to almost completely change the overall sex development of the organisms. They were able to, do this with SARI, SOX9, and with FGF9. So that's what these three experiments that you read in your book are all about, is just proofs of these three right here. So you don't have to read through all of those, but these are the proofs for these conclusions right here. So here's just kind of a layout. You have this in your book. It's a little different in your book. This is from an older version, but it's the same concept of how the initial stages of primary sex determination occur. So here are some of the universal genes. Included in that are SOX9, WNT, LIMS, SF1, and so on and so forth. So these are just kind of the genes that are necessary to get the intermediate mesoderm to start forming the bipotential gonad. However, after a number of weeks, after 7 weeks or so, then this is when these genes start kicking in, one or the other, depending upon whether you're XY or XX. So the SARI gene turns on the SOX9. The SOX9 will prevent Wnt and respondin and beta-catenin from being expressed. It'll turn on FGF9 and ultimately take on the path of the testes. However, if these are not here, then we'll show how in the ovaries, this is what happens. So in ovarian development, if there are 2 X chromosomes and no Y chromosome, then there's no SARI gene to turn on the SOX9 gene. So ultimately, the SOX9 gene gets turned off because Wnt and respondin, these are ovarian or X genes turned on by the X chromosome, that will shut down SOX9 gene expression. So that's what this is showing right here, is that Wnt4 and respondin, they actually turn off SOX9 gene expression. As a result, it allows for beta-catenin to be expressed. Remember, we talked about the beta-catenin and its role in transcription factors and the Wnt signaling pathway. So as beta-catenin is now able to turn on various genes, that's going to ultimately cause the bipotential gonad to become an ovary. Okay? A lot of these cells that initially would have become testes cords, instead of developing into cords and spermatogonium, now they're going to differentiate into a variety of different cells. So let's talk about what those are. So many of these cortical epithelial cells will differentiate into what we call granulosa cells and thecal cells. So what are these? These are the nursing cells, or the follicles, or the nursing cells for the or that form follicles that help in the development of the actual germline cells. So these granulosa cells and thecal cells that develop in the ovary, they'll surround every oocyte that develops, that will eventually undergo meiosis and form all the oogonium, and they're the ones that go through the process of maturing the germ cell. They secrete hormones, they respond, they ultimately set up the germ cell so that when ovulation occurs, it's ready to go. So these oogonium, they're the ones that enter into meiosis. These thecal cells and granulosa cells that will surround each oogonium are merely there to support it, to provide hormones, to maintain the cells, and the cells will actually enter into meiosis, the oogonium, before a woman's even born. Now they won't; they'll stop at a certain point, and in fact, even when the oogonium undergo ovulation, they're only at a particular. They're about metaphase 2, and it stops at metaphase 2 until the sperm fertilizes the egg. Once the sperm fertilizes the egg, it will finish meiosis 2 and get rid of the polar body. You guys remember your basic biology as far as polar bodies, or do I need to go over that again? Okay, so ultimately the oocyte, because of all of the maternal factors, what are the maternal factors again that are typically in an oocyte? Sequestered RNA, proteins, cytoskeletal arrangement, all those things that are necessary to get the genome started and cause fusion between the nuclei and get everything rolling. That's why the oocyte will actually not undergo cytokinesis. Spermatogonium will undergo meiosis and cytokinesis, in which you'll generate 4 sperm for every germ cell. But the oogonium, once they undergo meiosis 1, you end up having these haploid nuclei with 23 chromosomes plus their sister chromatids in each one of these nuclei. Well, after the first stage of meiosis, the oocyte will actually eject one of the nuclei, which we call a polar body. Then, as it goes through the second round of meiosis, or meiosis 2, it'll split up the sister chromatids. Again, you'll have 2 nuclei, each with 23 chromosomes that have undergone recombination previously in meiosis 1. And again, it'll eject one of the nuclei, which is also another polar body, only keeping 1 nuclei so that when the sperm enters in and that fuses, that you restore the full 46 chromosomes. In fact, as the sperm's entering in one way, this is on its way out the other way. That's what initiates the final stages of meiosis too. So the oogonium are undergoing meiosis before women are even born and all the way up to when the sperm finally fertilizes that oogonium. But for males, they just go through the process of just constantly producing sperm once sexual maturity hits. The follicle cells play an important role in hormonal secretions during pregnancy. What's the name of the thing? Corpus callosum? Is that right? Yeah. That ultimately, so they do play a role even after the oogonium has, or the oocyte has ovulated and left into the, for fertilization and implantation. So they do play a role in pregnancy and releasing various hormones even after the fact. They don't just completely degenerate right away, unless there is no pregnancy, and then they will. So here's just the major difference is instead of testes cores developing, you're going to have the formation of the granulosa cells and the thecal cells, and each one of them will surround an oogonium. And you're going to have millions of these. So women start out with several million oogonium. By the time they reach sexual maturity, probably only have 500,000 left. Most of them have degenerated by that point, and then they continue to degenerate during that reproductive cycle. And ultimately, when you get in your 40s or so and you run out of the oogonium, that's when menopause kicks in, and that's when you're done reproducing, essentially. So women have a short window of reproductive period. Men can almost produce sperm throughout their entire life. So most of us aren't reproducing in our 60s, but we can if we wanted to. Anyway, so ovarian pathway. Beta-catenin, this is a huge gene. It's the pro-ovarian development and anti-testes development. So the beta-catenin being turned on, it blocks SOX9 expression. Wnt and respondin work together to activate beta-catenin, and because SOX9 expression is not activated, then you don't get testes development. But then other genes get turned on, like DAX1 and such, that do push it down the ovarian development. Normally, if you don't have any SOX9 coming in to block any of this, this is the default pathway, so to speak. So whether you have, there are situations, very rare, where you get a form of non-disjunction, where you only get one X chromosome, that's still going to be female. Because even though there's only one X chromosome, since there isn't a Y chromosome, it'll still go down this process, although there's a huge mortality rate, like 99% mortality rate. It's called Turner syndrome if you only have one X chromosome. So it's not very common and always ends in infertility and whatnot. But it'll still develop female. So even if you have trisomy X or 3X chromosomes, it'll develop female. But if you have 2X chromosomes and a Y chromosome, which is Klinefelter syndrome, which is not hermaphroditism, we'll go over that today. Even if you have 2X chromosomes and a Y chromosome, you'll still develop as a male. Now, in some situations, males with 2X chromosomes actually have slightly underdeveloped testes, but some can actually be fairly normal. Another situation is an X and 2 Y chromosomes, but that, in fact, about 17% of men have an extra Y chromosome, and most of us don't even know about it unless we were to get our genotype done. It doesn't really hurt anything to have an extra Y chromosome. They've shown that it doesn't really do hardly anything else. There's so few genes, and most of these genes are just involved in initiating other genes, that it doesn't really screw things up. But 2X chromosomes and a Y chromosome, that starts to mess some things up sometimes, not all the time. And in fact, the same process that occurs in women also occurs in men with Klinefelter syndrome, in which one of the X chromosomes becomes inactivated. Now, X chromosome inactivation is related to hermaphroditism, but we'll talk later about how that occurs. But it's not the situation in Klinefelter syndrome that causes that. Yeah. So this is a key point in development, because after primary sex development, after either the testes or the ovaries develop, from there, that's where all of the secondary sex characteristics come about. So all primary sex, development is whether the bipotential gonad becomes the testes or the ovaries. That's it. These are the first things that have to develop before the other components undergo the development that is necessary. So let's look at what a sexually indifferent individual looks like. This is before the gonads become differentiated. You have things such as the Wolffian duct, the Müllerian duct. You can see here's where the kidneys are forming, and you have the ureter coming down to the urethra. So ultimately, in women, the urethra I'm trying to remember all of my parts here, are going to be separate. The urinary tract and the vaginal tract are separate, whereas in men, they're going to fuse together as far as the urinary tract and the reproductive tract. So here, let's say testes develop. What happens as a result? Well, one of the main hormones that gets secreted by the testes is testosterone. So testosterone starts binding to receptors that are present in the cells, and that allows various changes to occur. Number one is when it binds to cells in the Wolffian duct, it enables the Wolffian duct to survive. Now, the Wolffian duct makes some of the major portions of the male sperm delivery, such as the vas deferens, epididymis, and seminal vesicle. So that is what the derivative of the Wolffian duct is. It's going to fuse with the testes and the testes cords, so that when the sperm come out, they're essentially going to go through the prostate, out the urethra, and fuse with the urinary system. Okay? So it also causes, remember, SOX9 turns on anti-Müllerian factor, which is necessary to cause the Müllerian duct to degenerate. So this is what causes the Müllerian duct to go away, because the Müllerian duct develops into the female secondary sex characteristics. So let's look at that. If ovaries develop in primary sex development, they're going to secrete what? Estrogen, amongst other things, but estrogen is one of the major factors. And because they don't secrete testosterone, then the Wolffian duct is going to degenerate. And what are the derivatives of the Müllerian duct? It's the oviduct, where ovulated cells are going to go down through, the uterus, where implantation occurs for pregnancy, the cervix, between the uterus and the vagina, and the upper portion of the vagina. Okay? These are the derivatives of the Müllerian duct. So this will survive and ultimately develop, whereas the Wolffian duct will degenerate because there is no testosterone, and the Müllerian duct will survive because, remember, SOX9 is not turned on in ovary development, so anti-Müllerian factor is not initiated as a result.